from Micro TV, this is TWiEVO, This Week in Evolution, episode 103, recorded on June 26, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Great to see you. Great to be with you on TWiEVO 103. We're back on the live stream. It's been a minute since we've been together.
Some time, yeah. Been running around the world. And today, let's see, what's the weather here? You get nice weather there? It's 32°C here.
Yeah, it's been running hot here too. Almost hit triple digits on the old Fahrenheit scale in the last couple days, but coming down nicely by Friday. And we're no doubt we're into summer here.
But you could go up into the mountains and it would get cooler, right?
Well, I'm telling you what, that's the way to do it, is to, especially, it still gets hot during the day, but as you get into the mountains, even up 500 feet, 1,000 feet elevation, things really cool off in the evening in a beautiful way. That's the way to go. Yeah.
I've lost all of my textual stuff.
Oops.
Let me see if I can copy it. I'm going to lose you for a minute.
That's okay.
I'm going to copy it over. Let's see. How do I do that? Let's copy this. Let's say make available in all scenes. Try that. Micro TV. We got Venmo.
Yep, I can see it.
And now let's get back there. You can say, there you go. Look at magic.
There we go.
Do this, Nels.
Yeah, no, we're knocking the rust off a little bit. I think it's been just under 2 months. We're still right on schedule. Our one TWiEVO per month. It's just we hit the very beginning of May, and now we're hitting the end of June.
2 months. But really, it's been 2 months since we did a stream.
Yeah, just about. Wow. It's time flies. We're coming. I feel like I'm kind of sputtering to the end of the finish line of the academic season. I just did my last stint as a standing member of Genetic Variation and Evolution, a study section at the National Institutes of Health. My 4-year run has come to an end of reviewing grants. This has been a lot of work, but also a lot of fun, and I think a lot of really important conversations about how are we prioritizing the work of our colleagues, peers, what's getting funded by our taxpayer dollars here in a very competitive environment. And so anyway, between that, the kind of science, regularly scheduled science, traveling, and, as you go down the list, it's been a busy couple months, but it's good to be together.
I was on NIH study section for 11 years now.
Wow. As a single run, or kind of you signed right back up? How did that happen?
I can't remember. I know I was I was a regular reviewer for a while, and then the last 4 years I was chair.
Yep.
I don't remember, but I had enough. I mean, I get asked all the time to do other things, and I say, find other people. All right. I got other things to do, and I really would like to help you, but I feel like I've put my time in. Yeah. There have to be other people out there that can do it, right?
That's right. And, actually, this is it's so important that all of us at some point or another step up and do this, right? Because, this is really critical. It can feel like it's a ton of work. You're generally reviewing, and if you're a chair, you're looking at you're sort of organizing the meeting and looking at everything. But if you're even just a member, you're looking at between, 8 and a dozen applications, really wanting to get it right in terms of how, it's getting scored, et cetera. And then putting in a lot of effort to kind of have that conversation. But it's easy to say no, but I think as more of us say yes, that can really help keep sort of, the gears of science running in good ways. So, and I learned a ton doing it along the way.
You do learn a lot, yeah. Well, welcome everybody. I want to welcome, we have one moderator here today, Bart
Good.
UK. Thank you for coming.
Yeah, thank
really
you.
Appreciate it. And everyone else, tell us where you're from. I'd like to give you a shout out. There are 20 people watching, so say hello. Tell us where you're from before we get into the science here.
Yep,
John,
we're
I know,
running.
is from Minneapolis. Hello, John. Good to see you again.
Great. Welcome. Yeah, we're usually kind of sampling from around the globe. And that's reflects the work that we look at too in this week's or this month's paper that we're gonna consider in some detail, or reviewed preprint, I guess is actually what you would call it. We'll talk about that in a moment. Covers this. This is an international group.
Lex is from Lancaster, PA.
Welcome.
I wonder if being away for a long time, you lose people, Nels. What do you think?
Well, I think we just kind of scrambled the deck on the calendar. People were probably, ready for us to come in a couple weeks ago. And so,
And maybe Jeremy's from Seattle. Hello, Jeremy.
welcome. Good to see you.
And Claire is from the UK.
Welcome back.
Bryant is from BC with thunderstorms.
Ooh, yeah.
So I spent some time in Europe in the last few weeks. I was in Sweden and Finland for a variety of podcasts and meetings. It was a good time.
Very cool.
Good time of the Saudi Arabia. Here we go. We're pulling people out of the woodwork now, so you just have to ask them.
All points on the map. Great. Welcome. Good time to be in Scandinavia, Vincent, as the daylight increases and winter's grip
So first of all, the temperature was great. It was like low 70s, no humidity.
Ooh.
But also, it is very funny. At midnight, it was still kind of light out.
Mm-hmm. Oh, yeah.
And then the sun rose at like 1:30 a.m. So it never really got completely dark. It was very unusual, which is fine because, hotels have dark out, black curtains, shades and stuff like that. But it's kind of cool.
Very cool.
Pete's from Dorset, UK, driving over to his sister's for dinner. Well, enjoy dinner. You can always listen another time. Hey, MJ is from Austin, Texas.
Great.
Well, you and I were there not too long ago, Nels.
That's right.
I think it was back in March, right?
Yep, TWiEVO 100. That was a fun occasion.
Anouf has been following since 2000. Yeah, we got a lot of listeners for the pandemic.
2020, hopefully. Which pandemic are we talking about here?
It could be, yeah, I don't know, 2000. Not that I know, but 2009 would be H1N1.
Yeah.
2020, yeah, probably 2020. That's when a lot of people joined us.
SARS-2, I guess 2010 would be MERS, and was it 2000 that SARS-1 was on the radar?
2003, but not really pandemics, right? So
yeah. No, you're right. No.
Renzo from Ontario. Hello, Renzo. Good to see you.
Welcome.
Yes, so it's 2020, he meant. Yeah.
Yep. Well, you never know. There's a pandemic every few years, so you can.
It's okay.
Yeah.
There's pandemic. Even if you think this SARS-CoV-2 pandemic is over, there's still an HIV/AIDS pandemic ongoing.
Correct. Yep. Well, and have you been following as well the influenza? I forget is it an H5N1? I forget what it is in terms of.
It's an H5N1, yeah.
Epitopes, but moving from the avian populations to the cows, to cattle around the U.S. I've been watching that with a little bit of interest.
Barb says she brought in a new listener today, Lexa. Well, welcome, Lexa, and hope you enjoy. And thank you, Barb Mack, for that. Appreciate it.
Yeah, for sure. So, Vincent, we should dive right in to our paper here. So we're back in the game. This paper's title is a direct experimental test of Ohno's, OHNO, Ohno's hypothesis. And so we'll decode some of those details in a moment. But to get started, let's talk about the authors here. So this is, as I was hinting at, is what's known now as a reviewed preprint at the journal eLife, and we'll talk about that. But the first author here is Liliana Mahajlovic, and senior author Yolanda Scarley. Few folks here from, actually a few dots on the map in Europe. So Department of Fundamental Microbiology at the University of Lausanne in Switzerland. Some affiliations with the other institutions. My eyesight's going there. We've got your screen up, but it's
Too
a little
small.
bit zoomed out. We've got that's just fine. We've got the Santa Fe Institute. We've got Department of Oncology and the Ludwig Institute for cancer research, also in Switzerland. Institute for Evolution and Biodiversity at the University of Münster in Germany. Also, the Department of Evolutionary Biology and Environmental Studies at the University of Zurich in Switzerland as well. So, yeah, pretty interesting publishing model here. And I won't go, maybe I'll circle back depending on how we time out here to talk a little bit about eLife and some of the ups and downs there. But currently a really interesting publishing model. So the notion is that if you send in your manuscript, the editors there will do everything they can to try to mount a review of that. So they're trying to like lower the bar, sort of to filter it out. And then, as authors, you're meant to preprint it right away. That means make your manuscript available to the public. Then it goes through a peer review process. That's the reviewed part of the preprint. All of the kind of comments and the assessment by the reviewers, the editor, reviewing editor as well, are made public. And then at this stage, so the stage that we've found this manuscript is a version 1 reviewed preprint. So the author's first draft is up, the reviewer's comments are up, and then the reviewer, or the authors, I should say, are very welcome at this, or to do kind of what they think they should. So should they respond, they can respond to the kind of comments and criticism of the reviewers and update the manuscript, which is what happens for almost every scientific journal. But that's usually happening sort of behind the scenes before anything is made public or published. And then whatever the authors want to do, they can sort of declare it sort of the version of record, the VOR. So they could do that today, and it would just look as we find it at the open access website. Or they could review it, wait, and then say, okay, we've made these reviews, kind of went back and forth with made these changes, went back and forth with reviewers, and here's the updated version, or version 2, that becomes the version of record. So you can kind of watch that playing out in real time.
So Nels, I presume not every manuscript gets reviewed, but ones
No.
they think
Well,
are worthy, right?
Well, no. So that's the attempt here, is they would like to say, we're not going to make that worthiness sort of, judgment at the beginning. That runs into a practical problem, which is there's just too many manuscripts out there and too few reviewers if you need to get 3 of them to look at everything that comes in. And so that's where the model sort of hits the first roadblock, or the first kind of challenge, is that you end up having to triage at that that sort of early portion one way or another. The kind of interesting thing about this model too, right, is that so if you do go through the review process, you're committed to your work is already out there, and you're committed to the reviews being published online. So you get very negative reviews, or harsh reviews, or critical reviews saying, there's a fatal flaw in this work, and that might be kind of on the public record. So yeah, it's an interesting, it's an attempt to kind of dismantle the kind of possibility of sort of backroom judgments, or judging worthiness of something up front versus letting it sort of go through the process in a more transparent way, with some, I think, really cool innovations and advantages, but some really clear practical problems when you sort of think about the number of manuscripts out there versus the number of reviewers.
I guess we'd agree that peer review is essential, right? Because when something is published in peer review, then you have a lot of confidence that it's right? But
well, yeah,
the problem is there's not enough reviewers. Yes, for all the science that's put out there, and that's a big problem.
Yeah, exactly. And also, I think I agree with you. Peer review, in my personal experience, has been hugely positive in terms of
Mm-hmm.
getting feedback, advancing the work, sort of seeing blind spots, in some cases catching errors before they kind of got out there, which would have been sort of awkward scenarios down the road. Now, which I'm grateful for peer review for sort of advancing and strengthening the work. At the same time, peer review is not sort of flawless either. So and if you look at the trend
Sure.
lines of the number of peer-reviewed articles that are then later, retracted, that's not a small number. And sometimes the most, splashy of papers are the ones in sort of hindsight that ended up being, a little bit too splashy in terms of what's the real kind of rigor or the evidence behind it, or how reproducible is that evidence. So yeah, it's kind of a mixed bag. And so eLife, I think, has really been trying to innovate in this space, in some ways really effectively. In other ways, has gotten kind of tied up into some kind of controversial moments as well.
Sure.
Yeah. And so anyway.
Hey, but Dales, before you go on, we have two moderators joining us.
Oh,
We have
great. Fantastic.
Vanity Nutrition is here. Thank you for
Yeah.
moderating. Appreciate it. And
Welcome.
Tom is here.
Oh,
Welcome,
good.
Tom.
Fantastic.
Thanks for
Good to
joining
have you
us.
all back. Yeah. As Vincent and I were saying, we were kind of scrambled our calendar a little bit here. And so glad everyone is finding us here in the real-time live stream. Of course, this will also be available as a recording going forward, for those of you with patience and fortitude to, to watch Vincent and I kick around some science. Okay, but so regardless of publishing model, here we are with a really interesting paper or reviewed preprint called A Direct Experimental Test of Ohno's Hypothesis. So what is Ohno's hypothesis, and who is Susumu Ohno in the first place?
Oh, I
So
thought it was Yoko.
yeah, that we'll revisit that for the great title that Vincent that you came up with for our episode when that gets posted. So it's not Yoko Ono, but Susumu Ohno, who is a giant in the field of molecular evolution. So both evolution and also genetics, including cytogenetics. So he passed away, I think about 24 or 25 years ago. He'd be almost 100 years old today. But he is famous for championing the idea that there's really a central role of gene duplication in the process of evolution. And so, when we think about sort of, genetic innovations or genetic diversity, and certainly at the time when he was, actively practicing molecular evolution, the point mutation was really, and continues to be, maybe sort of one of the dominant mechanism that we think about when we think about evolutionary change. And what Ohno brought to the table was the idea that it's really like evolution happening in not just letters, but in full sentences or words here. I don't know the right analogy, but where genes duplicate, and then this becomes the raw material that natural selection is acting on. And so under hiding in this sort of idea is the concept of what we call neo-functionalization in the field. It sort of has a kind of means what it says, that new functions become possible or are facilitated by gene duplication events. That is the copying of one gene into two. And that can be mediated by a number of sort of genetic processes. And Ohno, Susumu Ohno, wrote the book. It's called Evolution by Gene Duplication, to really advance this idea that still has echoes or ripples in the field of molecular evolution today. He also, some kind of notable cytogenetics here. So he discovered or put together the idea that the bar body that was seen as people were starting to peer at chromosomes under high power microscopes is the condensed X chromosome. That was, he pointed that out in the mid 1950s. And so there's all these great sort of historical references back to Ohno's work. So Ohno's hypothesis, which we'll be talking about today, that gene duplications or duplicated genes, because you have a duplication, it can kind of cover for the other one, allowing the other one to mutate and be acted on by selection. And the hypothesis is that this would speed up the evolutionary process. And so we'll unpack that idea. This paper attempts to test that idea directly, as pointed out in the title. But there's also some other, fun connections here. So Ohno's law harkens back to the X chromosome, that it's conserved among mammals, and that the genes on the X chromosome are constrained or conserved. That conserved. That was later coined Ohno's law. And there's also Ohno's dilemma, which is, how do duplicated genes persist? You have this extra copy, and because it's covered by the other one, if you get a mutation that disables one copy, the other one's there to kind of cover it. And so, how is it that this neofunctionalization can happen if it's a lot easier to lose something than it is to sort of have something persist? And so,
Nels, don't you want to
yeah.
have Nels' dilemma someday?
Well, okay. So I'm glad you raised this point, Vincent, because as the whole part of the reason we started this podcast some years ago now was to kind of really try to highlight what's happening in evolutionary biology, sort of not in this era, where experimental approaches, as we'll see in this paper, collide with sort of the ideas or the arguments. And so I would say, if I was looking back historically, that I think evolutionary biology has suffered a little bit from kind of the fact that there are all of these giants in the field. And don't get me wrong, I think there's a lot of really great characters and interesting and science is practiced by people. And so I don't get me wrong, I love to celebrate the history and the scientists and who they are as people. But when we merge that together with the science itself, it can create some issues, which is if it becomes about kind of personalities and arguments versus data, we can get a little off the rails sometimes, or kind of gaze at ideas that might have been, someone said something 20 years ago, and I'm going to dedicate my research program to that. A little bit of a blinking red light. I think one of the totally exciting, great things about the field of evolutionary genetics today is that we have all these experimentalists kind of coming from all these different backgrounds from all over the map, bringing that sort of hypothesis driven or even comparative, just like genome scale, what does the data look like, even as you do comparisons or experiments. And so it becomes less about the argument or honestly, the sort of old middle-aged white guy syndrome, and it becomes a lot more about what's the data and what's going on. And the more science is litigated there, I think the more progress possibly can happen. So anyway, sorry, a little bit of a
no Nels' dilemma. That's fine.
So no.
But you have the kind of name that lends itself to it because it's very short,
Yeah.
Ohno, Nels, and so forth.
Yeah.
Nels, can you give us an example? And we've talked about this a lot on TWiEVO. Can you give us an example of a gene duplication where we know what the original was and the duplicated with a different function?
Yeah. I mean, so this is so gene duplications are happening constantly across the board. I mean, so I'm, and I'm going to, just as I'm trying to put myself to the side, I want to bring in a little bit of my own work here.
Right.
Because we've been thinking about gene duplications. Part of the reason I thought it'd be really fun for us to grapple with this paper, we've been looking at gene duplications in virus genomes. I did this as I was opening my lab and have been seeing some interesting patterns there. So in this case, the gene duplication, so it involves a gene that the virus gained from the host genome. The gene is called K3L, it's some alphabet soup. It gains copy number in virus genomes, and then because of that, evolves in interesting ways. Now, in that case, where we've been studying, it's not just 2 copies. It can be 10 copies, 20 copies. And virus genomes probably have some flexibility to do that. We're going to be talking about bacterial genomes as it relates to this paper. But of course, we and other evolutionists are super interested in what does gene copy number look like in our own genomes? And the answer there is actually, there's quite a bit. So paralogs, these are, copies of genes sort of in the same family. These were ancient duplication events, and these are scattered throughout the genome and probably enriched to some degree for genes that encode immune functions, for example. And so the history of this is long and wide. I would say, and we'll kind of get into this with the paper, the idea that there's just one copy, so from 1 to 2, and then you just stay at 2, those examples I'm sure are out there, but are probably less frequent than the more messy and kind of chaotic evolution. And part of the reason for that, why things get messy and chaotic, is recombination. Once you have 2 copies of something, you have sequence homology, the ability to sort of mix and match through recombination really goes to town, and that can quickly lead to more copies. even potentially in our own genomes that are not quite as flexible or quote unquote breathable as some of the microbial genomes out there.
We should also mention it doesn't have to be a coding sequence. It could be a non-coding. It could be an intron. Could be regulatory sequences, right? All of that can be
Abs-
duplicated.
Absolutely. Repetitive sequences. I mean, one of the
Yeah.
famous repetitive sequences, of course, is the telomeric repeats. So these are 7 base pair repeats that you find at the ends of our chromosomes. And they continue to sort of grow, like kind of grow and erode through chromosome as cells are dividing, and the ends of the chromosome will naturally be shorter. Telomerase catalyzes the addition of these 7 base pair repeats to keep the chromosome ends from sort of melting away before your eyes. And so, yeah, all kinds of duplicated sequences, repetitive, non-coding, as you're pointing out, and coding genes as well, which is the topic of today's paper. So the notion here, kind of, if we simplify it and just think of, and that's what we'll do here, is we'll take a gene in one copy and compare that in real time experimentally with a gene in 2 copies. There's several fates for a duplicated gene. So you imagine starting with one, a duplication event happens, and the mechanics of that at the moment aren't super important. The gene could be lost. You have 2 copies. You go back to one. That sort of hints at Ohno's dilemma. That was coined, by the way, by some Scandinavians from the neck of the woods where you were in your travels, Vincent. So Ulfar Bengthorsson, Dan Anderson, and John Roth, I think, were the ones who coined Ohno's dilemma, maybe coming up on 20 years ago. Another fate for these genes is both copies persist. So as soon as you get a second copy, depending on the gene regulation here, you now have, twice as much of that gene being transcribed or made, which means twice as much of that protein. Yeah, it can, it might be somewhere between 1 and 2, or depending on how the regulation is working, might be a different number. Here, actually, I think they point out, so for the gene, we'll get into the details, but the gene that they duplicate, they get 1.5x gene dosage. So immediately, a duplication event can have, potentially consequential impact, which is you've now turned up the dial on how much of that protein, that whatever that gene is encoding, is in the cytoplasm of
It's
the
interesting. It's
cell.
not 2x, right? Because they're 2 copies. So in addition, there's some regulation going on. Clearly, right?
Absolutely right. Yeah. The third sort of fate or possibility that's been pointed out repeatedly is the partial loss of function or subfunctionalization. So imagine a gene encodes 2 jobs, and this is not unusual at all. If you have 2 copies, one, and you come back a million years later or something like that, you sort of a division of labor, genetic labor might be set up where a copy, the original copy, so to speak, parental copy, is doing one of those jobs, through genetic divergence and selection, and the second copy is doing a different job. It's specialized. And I think, that's kind of intuitive to cell biologists who think about how, as you look at gene families expanding and, cells conducting what feels like more sort of complicated jobs, with compartmentalizations, transitions from the prokaryotic to the eukaryotic world, et cetera. Kind of intuitively, it could feel like that some of the gene duplication could lead to specialization, subfunctionalization. So this can also spill over as you go from a single cell, which we'll be doing today with E. coli, but to multicellular life, right? Now you can have expression of one gene in one tissue. So all the cells encode both copies of the gene, but you just turn it on in one tissue type, one of the genes, you turn on a different one in a different tissue type. That could be a sort of a form of specialization here as well.
So
Um,
Nels, when we
yeah,
talk about this,
yeah.
this will happen in an individual organism, and then if it's advantageous, it will propagate. So we're talking about many, many years for this to occur, right? It doesn't happen overnight.
Oh yeah, absolutely. And so, that's all driven by, so there's this sort of, I think, and it will kind of end up here that the duplication event itself is a really fundamental, kind of impactful genetic change. But then depending on population size, generation time, mutation rates, that kind of will dictate then how long these sort of, fates will be determined. But you're absolutely right, Vincent. So I think, for microbial populations, viruses, this could be happening relatively, from an evolutionary standpoint, kind of in the blink of an eye over, maybe months or years. For other populations, or generations, like our own species, right, or other mammals. Here it could be, on the order of hundreds of thousands of years or millions of years, depending on what's happening here. Although, with,
but on a
yeah,
daily basis, say a
yeah,
human, are
yeah.
we undergoing
Oh,
duplication right now, you think?
well, someone is. There with enough people, so population
Yeah, yeah.
sizes, right?
Okay.
But
Okay.
whether that gets selected on,
Right. Of
that's the
course.
process that you're pointing out is going to take a really long
Of
time. Yeah, yeah,
thousands and thousands for humans, many, many thousands of years, right? Yeah.
yeah, absolutely. Yeah, yeah, yeah. Um, but never underestimate the speed of evolution. I mean, I don't know how to, yeah. Anyway, so that, but
That's a
uh,
good saying. I like that.
yeah, I try to remind myself of that kind of as I go here. And then the final fate is these two genes after the copying event or the duplication event, the genes diverge and evolve in the second copy, so to speak, evolves a totally new function. So the first one is doing, it might even have multiple functions, but it's doing both. And
Mm.
then this copy is now doing an altogether different function. It might be related, but it's not being performed or covered by that first copy. And so that's really, I think that notion of neo-functionalization has really captured the imagination of a lot of scientists, because that sort of gets at this, how do you adapt? How do as a population goes to a new environment or is faced with a new environmental insult, a new pandemic or something like that, how do you survive? And it's in that copy that might gain a mutation that gives you resistance to this viral disease, and that's a neo-functionalization event. And that now we can see selection acting, right? Everyone else that doesn't have that duplication or that mutation, that diverged copy, is vulnerable and might get wiped out, might not have kids, and very quickly that new function would sweep to fixation. That would be, who's here today. And in fact, I think, there's some real good evidence out there that the fact that we're here, we kind of stand on the shoulders of our ancestors, that all of these genetic events happened and were litigated. And, we were the beneficiaries of that. We're the lucky few who are here today, because of a lot of neo-functionalization.
Mm.
Okay, so the last point here, I think, is just kind of in terms of the definitions, is this notion of, and kind of obvious to this, is the notion of genetic redundancy. As soon as you have a second copy, you're redundant, right? They're identical to each other. This is also referred to as robustness, from a genetic standpoint, or mutational robustness. And so that's the thing that the authors would really like to kind of get at here with their experimental system. If you have two copies of a gene versus one, are you more robust to mutations because of this redundancy? And then getting to Ohno's hypothesis, do you evolve faster, meaning that you hit a phenotype, they'll pick a phenotype just from an experimental standpoint, and we'll talk about that. But do you get there faster on average if you have two copies versus one copy? And spoiler alert, the answer for this system is no, you don't. But there are some interesting kind of dynamics here that I think start to unpack some of the ideas or arguments tracing back to Ohno, and putting it into this experimental light. Okay, so back to the dilemma, Vincent. Adaptive mutations or beneficial ones are much more rare than deleterious ones. It's a lot easier to break something than it is to fix something or to build something new. And so the dilemma is, do you have enough time? You've duplicated this thing, but now as mutations are happening, you'll just lose one or the other, and you'll be okay because of that redundancy. And then you can't really come up with a new function or create something new. Evolution can't be creative because you sort of lose this thing. So there have been, that's been grappled with now for decades by molecular evolutionists, and alternate models have tried to solve the dilemma. The one that these Scandinavians, Bang Thorson, Anderson, and Roth have put forward is this IAD model: innovation, more alphabet soup, innovation, amplification, divergence. And here, in order to solve the dilemma, they take away the constraint of just having two copies, where you can go to 4 copies, 8 copies, 16 copies. And so, there you're sort of, the redundancy might outpace, basically, you start to balance out the rareness of a beneficial mutation with the frequency of a deleterious one by not just having 2 copies, but many, which I love this idea. And so this actually, we've been working with in viruses, the pox viruses, and identifying patterns that sort of fit this innovation, amplification, and divergence model, where you don't just have 2 copies, you quickly have like 20 copies, and seeing how viruses can evolve right before our eyes with that sort of dynamic. So here, and to get at that, what we called the gene accordion, we did experimental evolution with vaccineovirus, the vaccine strain of pox viruses. So this is a pandemic smallpox, the one that we eradicated using vaccination successfully, and should be doing more of with other viruses, as we're on the cusp with poliovirus and as we're sort of grappling with these other pandemics, and if anything, in some corners, letting go of vaccines, and I think really sort of messy ways. But to stay on topic here, so they're using experimental evolution, or a flavor of it called directed evolution in E. coli, to try to get at some of Ohno's hypothesis. And here they really want to keep the system at 2 copies to kind of get at those like original ideas. And they have set up the system so that that recombination isn't possible. And that's like, it's a clever engineering scheme that really lets them sort of distill the genetics here. But also, with all of kind of experimental evolution, where you set up a system to try to provoke evolution, oftentimes in microbial populations, there are trade-offs that you're making. How natural is the system versus engineered? How much of what you observe in the laboratory or in the test tube reflects what's in the wild? And I think, they're a little bit vulnerable to that criticism. So, but I think from some of the other alternate models related to Ohno's hypothesis, this is sort of like evolving with one hand behind your back if you can't do recombination. I think that's what solves a lot, or gives sort of the flexibility of duplicated gene systems to do a lot of interesting evolution. So anyway, let's get to the details. So how do they do this? So here they want to have a system where they have in one population 1 copy of a gene, in the other population 2 copies of the gene, allow mutation and selection, or put selection onto a population and see if there are these differences in sort of these key parameters of how these copies of the genes evolve. So the gene they picked is a green fluorescent protein, or GFP. But this one has a cool property. So it's I forget the critter it was taken from, but a lot of these marine animals, right, encode these fluorescent proteins of different colors. If you excite them with UV light at a certain wavelength, they emit green light, or in this case, both green and blue light. And so they have the ability, they don't really take advantage of it for this first paper, but they have the ability to select on both green and blue, or both, or one at the expense of others, and can really tease apart some of the constraints on the system. We're just going to focus on green for simplicity, and since that's what they did, they were kind of highlighted in this first paper to get at the questions related to what happens with the quote-unquote speed of evolution if you have 1 or 2 copies. Okay, so basically what they do is start the experiment with a version of this GFP, or co-GFP as it's called, that is emitting equal amounts of green and blue light, and then they want to see what happens. And so the way they control the system is they put the 2 copies of this gene, GFP, identical copies, onto a plasmid. So this is just like an extra piece of DNA that they can put into E. coli, into bacteria, and the E. coli will maintain and replicate that plasmid. It's not in the chromosome or in the genome, but it will allows them to like quickly engineer and sort of change the system in real time. And what they did to block recombination is they put the copies of the GFP gene in opposite orientations so that it can't sort of, just the mechanics of recombination, it can't match up. And so it holds it at 2 copies. And then they can induce, so getting at your point from earlier, gene regulation, they can induce it with 2 different chemicals. And so they can actually turn on both copies or one or the other. And that actually is a really cool kind of engineering trick that will come into play here in a few minutes. So then, to set up 2 copies versus 1 copy, so both populations will have that plasmid with both copies on there. But in the 1 copy case, they've disabled the gene with 3 mutations, 3 deleterious mutations. And so in essence, it has the same kind of cargo on the plasmid, or the piece of DNA that's in there. But it only has 1 working copy versus 2 working copies at the start of the experiment. And so, that helps to control for things like the size of the plasmid, other sort of parameters, as they want to test the differences, the mutations that arise, and what that means for the phenotype, which is the green light that the GFP is emitting. And so, and because they have 3 mutations, the notion is you're not going to revert with, you're not going to hit the jackpot of 3 mutations in sort of a short evolutionary time course to all of a sudden have 2 working copies again. And then the final little tweak here, Vincent, is like the difference between experimental evolution, where you let mutations accumulate, you let the E. coli generate the mutations for you as they're just replicating, as the cells are replicating and replicating the genetic material, the DNA, mutations will be introduced. They want to kind of put their, thumb on the scale to make mutations happen faster. So what they do is they take the plasmids out of the E. coli, or if they're in there, they do a technique. In this case, isothermal DNA amplification, which is a mutagenic process of amplifying the plasmid. And so basically they can get 2 mutations that will change 2 amino acids in the protein encoded by the GFP gene per round, 2 mutations per round of their experimental scheme. And so they're doing all the mutations in vitro, so to speak, outside of the cells or without the aid of the cells, but then they put the plasmids back into the E. coli, select on ones. In this case, the selection will be how much green light or blue light, or combos of both, the cells are emitting. And then they collect those plasmids, pull it out, do that same process, wash, rinse, and repeat. And that's directed evolution. So you introduce mutations at each round, then select on a phenotype, in this case, how much green light the cells are producing, to test their predictions about how evolution will unfold in the 1-copy gene versus the 2-copy gene populations.
So they're imposing their artificial selection, obviously, because this has no functional activity in the E. coli, right?
Exactly right. And so the way this works is they use a machine, a FACS machine, or fluorescence activated cell sorter. So what this machine does is and it's pretty impressive, right? So in real time, you shoot single E. coli cells through it. It reads how much fluorescence that cell is producing, green or blue. And then if it passes a threshold, it saves that cell. It squirts it into the well of a plate, and you can then recover that. If it doesn't emit enough green light, or it doesn't pass that threshold, then it gets thrown away. And so that's human-induced anthropomorphic selection, or FACS-induced selection happening. Mm-hmm. And so most of the cells, right, that are in that population are going to get flushed down the drain, and it's just those rare ones that pass that threshold of this is getting more green, let's save it. And so that's really the game here of selection, which is the key to then directing the evolution and seeing these patterns move forward, and to do it quickly. So they just do a few generations, a few rounds, something like, we're looking at the first 5 rounds of mutation and selection of this directed evolution scheme, and they already see differences, which makes it accessible to the experimentalist. You don't have to wait a million years and then come back and look what happened. This happens really quickly because it's such strong selection using the FACS sorter, the fluorescence activated cell sorter machine. Okay, so all kinds of possible quick complexity, like you could argue, or you could, however you want to slice and dice your selection. What they end up doing is, for this paper, is just focusing on green, but you could select on green and blue, or you could select on ones that are green but not blue, and really sort of push the system farther. I think for this, kind of the first questions they're asking, they kind of saw the same results, whether they selected on green and blue, just green, just blue, etc. And so let's, now that we have the system set up, let's just kind of march through the questions that relate to Ohno's hypothesis. So the first question, are having 2 copies, does that make you more robust to mutations? And the answer is yes. So this is shown in figure 2 of the paper. And basically what they're looking at here is as they recover their cells after each course of directed, or each round of directed evolution, which they're calling generations, they ask what fraction of these cells are fluorescent. And if it's the 2 copy, so if right at the beginning, after the first round, if it's the 2 copy population, about 40% are fluorescing green, or producing green cells, and only 28% are in the single copy. So thanks for putting up. So the single copy is blue, population double copy is red. And so you can see it like by generation 2, it's like crashing out. Because you only have a single copy and you're introducing all these mutations, most of which will be deleterious, will ruin the gene function. You can see that it's sort of, starting to scrape the bottom of the barrel for the number of cells that can still produce the GFP or can still fluoresce green as you're selecting them. But what's interesting here, right, is that by generation 4 or 5, both populations are recovering. And this is, hinting at the selection that's happening here. The farther you go in the experiment, and as the fraction of fluorescent cells that are coming out each time increases, what that represents is that those mutations that are actually beneficial are coming up in frequency. And so that's sort of like balancing this out. And so the advantage, so to speak, or the redundancy, I should say, that you have early in that with 2 copies is lost. That window is closing as selection is starting to be imposed there.
Nels, why is that happening? I'm not quite sure I understand why those curves come together at some point.
Oh yeah, so basically, I guess you could think about it that maybe one way of thinking about it is the difference between a single individual in a population and a population dynamic. So if you're a single individual with 2 copies, right, and there's just mutations happening, one is likely to get knocked out, but the other one is likely to persist, right? Whereas if you only have one and you have that mutation, now you're off the charts, right? Yep. Right. But then as you continue to select on this now, just from a population level, the number of these guys that are intact or have a working copy. So they might still have mutations, by the way. They probably do. Actually, the authors don't look at this or really focus on this. They're focusing on the mutations that cause amino acid differences. There's also all of these. There'll be plenty of mutations where it encodes the exact same gene, and so pretty soon in that even that single copy population, the ones that remain will have just as many mutations, but they're ones that aren't disabling the gene, right? Because those are the only ones that can come through. And so as you sort of, go farther, basically, those are the ones that persist at the population level. The other part of this too is that even if you have 2 copies, one gets disabled, and actually they'll look at this and use their system in a clever way in the next set of questions they ask. But not only at some point, not only do you lose that first copy, but you start to lose the second copy as well. If you're walking around and with one working copy, that's vulnerable to being disabled as well. So I think that process of robustness is working kind of in both directions in this case. Yeah. Okay. So then, so that kind of gets at robustness, which is pretty intuitive and makes sense. The real kind of heart of Ohno's hypothesis is asking, are the 2 copy populations evolving faster? And faster is like a tricky term when we're thinking about evolution, because what do you mean by faster or better, or it's sort of relative. Here they're defining that phenotypically as producing more intense green fluorescence. And do you get to that more intense green fluorescence faster? That's sort of how this is being framed. And the reason that would be, sort of adaptive in this case is that the FACS sorting, you're setting that threshold for what qualifies as green. It gets increasingly stringent. So you start with the threshold is pretty low. If you're just sort of, even maybe green fluorescence isn't totally lost, but there's a mutation where your half is fluorescent, those ones still come through at the beginning of the experiment. But then as you go generation to generation, they sort of raise the bar. It has to be a really green cell the farther you go. And so the question is, do the 2 copy ones get to that bar faster? And the answer is no, they're about the same. And so this is in figure 3. And so similar sort of curve, but on the y-axis instead of fraction of fluorescence, it's a different measure here. It's the amount, the log 10, or the amount of green fluorescence. So here you can see that, both the red line, the double copy population, and the blue line are much closer at the beginning and throughout. And in both cases here. And so this is actually sort of, fires a blow against Ohno's hypothesis. But what they do note is that there's more variance within the genes that encode that higher green fluorescence.
Mm-hmm.
If you have there's more diversity in the population. And so I think this is important to kind of, as we're weighing this work, is, to not take all of the results or not to sort of generalize all of the results from one system into, like, this is how it's working across the board. And so, having more diversity in a population from the two copies, that could be super beneficial. It turned out, and this is kind of jumping ahead a little bit, but as they looked at the genetic basis of, increased green fluorescence through the course of the experiment, they could get there pretty in a simple, like, one mutational step, or at the most even 2 steps or 3 steps. A lot of mutational processes, or for, finding, basically for evolution, natural selection, finding beneficial functions, could go through much more complicated processes where there's sort of contingencies. If you get this mutation, then the second mutation could work. But if the second mutation comes first, you don't get to the first, or, the first one doesn't matter. And so because there's more diversity in that population, a more complicated sort of so-called epistatic trajectory or mutational process where selection is acting might be facilitated by the 2 copy in ways that it's more constrained in the single copy. So in that sense, depending on the function, maybe Ohno's hypothesis does hold up. But for this case and the experimental test, the answer was sort of a resounding nope. It's about the same. So then, they can get at some of the other questions. What about the dilemma? How much loss of function occurs with the 2 copy? And so here, because they can induce one or the other, they can ask, so does that, for whatever cell has both the copies to begin with, do they, as they go through selection, is it because they have 2 working copies, 1 working copy? Which one is it? And so in order to test this, what they do is, so up until now, they've been inducing with both the chemicals, so turning on copy 1 and copy 2. But if they just do one of the chemicals that induces the gene, now they can just turn on copy 1, or use a different chemical, just turn on copy 2, and then ask, okay, if I turn on 1, are you a green cell? If I turn on 2, are you a green cell? Or if I turn on either, are you a green cell? Or, or not at all. And so what they find here is that most of those 2 copy situations, the farther they go in the experiment, it's only 1 that's covering, or that's encoding that function. So I think after the first round of mutation, 30% of the cells that they test, that they induce one or the other, the clones of them, are depending just on 1 copy. So 1 copy is inactive in 30%. And then by the end of the experiment, 4 generations, 80% have 1 inactivated copy. They're depending on the other copy. And so that's the dilemma, right? It's sort of this window of opportunity where if you probably come back after generation 5, 6, 7, it's like 99% are just, depending on that 1 copy, and that other one is disabled. In which case, either it would have to go through a mutational scenario where it reverts or finds a different function, neofunctionalized. But that gets hard. The farther you go, the harder that gets, because there are these disabling mutations that take away, that enzymatic function in that case.
And this is
So,
after
yeah.
they take the plasmids and mutate them and put them back in, right?
Yes. And so the more you mutate them and put them back in, the more times you lose 1 copy. And here they can quantify it down to, right down to the percentage. And to see it go from 30% to 80% in the course of just 3 rounds of mutation is pretty wild how fast, with 2 amino acid changing mutations. I mean, this is sort of getting at the reason we call mutations. We think of them as deleterious or harmful, in this case, in terms of the enzyme function, which is to make the cells glow green.
So the mutational robustness that we saw before is looking at a population with both copies, right? And so
That's
most
right.
likely it's going to be just one or the other that's giving you that robustness.
Well, or another way of saying it is, by 4 generations, there's 20% that still have 2 working copies.
Yeah.
And those ones are still robust, but it's like diminishing robustness the longer you go, basically. Yeah.
That's what we saw in that robustness experiment too. After so many generations, there's no advantage of having multiple copies.
Yeah, that's right. Exactly. Okay, so yeah, um, so the single copies catch up, um, and then but they do so. But there's more going on under the hood here, and so we already mentioned this. So there's more, or the authors already mentioned this, that there's more genetic diversification here. So how do they measure that? So that's what they kind of do next, is they ask about are they diversifying more rapidly? And here the answer appears to be yes. So here in the way so diversifying rapidly versus going faster are two different things here, which it gets a little bit confusing because it sounds like we're kind of all pulling in the same direction. But so in the way they test how they're diversifying is by measuring a ratio, the DN to DS ratio. So we've talked about this before on TWiEVO. So this is like most of a lot of evolutionary biologists use this. You'd compare as a comparative approach to ask the sort of flavor of the mutations that are accumulating by comparing these genes. So what they can ask from within their populations from the beginning of the experiment to the end is how many of these mutations that they see by the end are ones that change the amino acid residue, a so-called non-synonymous mutation, the rate of that divided by the rate of synonymous or mutations that don't change the amino acid. And so what they see is that number, that ratio is higher for the ones that have two copies versus the ones that have one. And this is probably really getting at the constraint that you have in place. If you only have one copy, you can't lose that one copy, or you won't make it through that FACS sorter. You'll get thrown out. And so that means you, as a population, you start to see more of these synonymous or quote unquote silent mutations. It's a DNA change, but it doesn't change the amino acid. And those are accumulating at a higher rate in the one copy because you can't lose it. Whereas there's less constraint. You can tolerate more diversity. DNDS is higher in the two copy ones because let's say you have a non-synonymous change, but you have that sort of intact copy covering for you, like you can get away with that. And so the DNDS ratio in the population is elevated. Really cool, I think, to see that sort of empirically, even over the course of this short directed evolution situation. Okay, so what are these mutations? So they're, we've kind of been picking at the edges here of the rates and the frequency of debilitating mutation, but what are the mutations that allow the cells to become more green? And they put their thumb on 3. So these are, we're into alphabet city zone here. Here the letters are the amino acids, and the number is the amino acid position within the protein. So the one mutation that sweeps very quickly to fixation is they start at position 147, starts as a glycine amino acid, gets a DNA mutation that encodes instead for a serine. And the mystery here is that, well, if you just take the enzymes outside of the cells and ask and hit them with light, how green is it? The answer is they're the same. But if you take these genes with the different mutations in E. coli, the answer is the serine substitution is more green. And that's because it's expressed at a higher level. And so again, in this case, we're not changing the function of the enzyme itself, but how much of it is there. And then there's 2 other mutations that show up that do kind of influence the structure. Here, there's a valine that changes to an aspartic acid. There's a leucine that changes to a methionine. And so they can go, first of all, they just confirm that. They go back and make those mutations separate from any other, the background mutations that are the silent mutations, et cetera. And they show that if you make those mutants, they're and express them in E. coli, they're more green than if you have the ancestral or the parental sequence. And then they can ask, okay, these are the mutations they think are responsible for these things becoming more green. And do they arise earlier in the copy 1 versus copy 2? Meaning if you have 2 copies, you get to that, it's sort of, again, asking, are you like evolving faster? Do you get to that beneficial state? And the trend is that they seem to appear a little bit earlier in the 2 copy system, but that not really. It's pretty subtle, and you could kind of explain that just with sort of, technical reasons with the experimental setup versus evolution is really sort of has a leg up in the 2 copy scenario. Not to mention, so again, like this system is a bit artificial. There's no recombination. So if you have recombination, what can happen is, let's say there's 2 or 3 beneficial mutations, as they've sort of outlined here. And these are, if you can undergo recombination, let's say you have the 2 copy, it shows up in copy 1. One of those beneficial is in copy 1, another is in copy 2. Those can quickly recombine into a version that you don't have to get that hit on that mutation by chance again. You just sort of bring together those 2 sequences through recombination, and that would, allow the 2 copy to move faster. I think my intuition is that's what's happening in these more natural systems where you haven't taken recombination off of the table, but they really wanted to just kind of keep that clean for the experiment that they set up here.
I don't know, maybe useful to, they throw away the ones that are not green, but maybe it would be useful to look at some of those to see the frequencies of deleterious mutations, 1 versus 2 copies,
Yeah, I agree. And there's no shortage of those. So that's like
Yeah.
that's the thing with evolution is the thing we see is the rare thing that persists because selection is so strong. But yeah, no, I agree with you, Vincent. It's, what is the denominator here, or the comparison point? And there's so much diversity out there, to draw like, something meaningful. Could be a little bit challenging, but you're right. That's part of the deal here, that we often never consider or see because it's lost sort of in the evolutionary record. Yeah. So
it's kind of interesting that we don't see more amino acid changes leading to higher green. It's just a few, right?
yeah. Oh yeah, exactly. And I think
Kind of
that,
surprised at that.
I agree. That speaks a little bit to, I mean, that would be fun to, so the field of fluorescent protein biology is, there's a lot of action there, in part because you'd love to have more colors. This one's really cool because it does both green and blue. There's also the whole kind of spectrum, whole rainbow of fluorescent colors, and all kinds of directed evolution to get ones that are sort of emitting in distinct wavelengths, because the cell biologist would love that, because every time you hit one, you get another color channel.
I bet they're in the ones that they throw away.
Yeah. Right? Oh, yeah. So, and so there you could
that make other colors, because they're just looking for green. But I bet in the pot with no green, there's red and yellow and so forth, just happening randomly. Why not, right? Because it only takes 1 amino acid change at the active site to make a different color.
Yeah. And so there, and you could also select on that, right? So the facts, you can kind of set that bar. So that would be a fun way, of what everything you throw away, you could also select differently there as well.
Because that's where, if we're looking for new functions, that would be a new color would be a new function, really.
Absolutely. And so you're like, that's another, I think, really important part of the conversation here is, as experimentalists, how we define a new function. Is it increased green? And is that just because there's more expression? So that's another sort of take home, I would say, from this system, is that the fundamental advance was just when they duplicated. So now they've got 1.5x. So that, the speed came there in terms of, or the evolution was fast when that second one came online. It increased the expression. Another point mutation shows up, increases the expression from a single copy level. But that's really, kind of the gears of evolution here are less about that unicorn, the horse getting a horn and becoming a unicorn, but more about the horse just growing a little bit bigger. And so, your point's really important, Vincent, because when we talk about neofunctionalization, like, what are we testing for and how frequent or rare might that be? This experimental system is like, again, like not even a blink of an eye of evolution, right? It's just 4 generations. And so, the patience to see this through for a year or 2 years or 5 years, where some more exotic evolutionary pathways, which would be closer to, I think, what most people would sort of, put their money down for, oh, that is a new function, versus here where it's sort of defined pretty modestly, I would say. Yeah. But even here, so, more robustness, some signs of that, more diversity, but not faster, in the sense that there's, that the green intensity rises faster in the 2 versus the 1. I think the caveats here again are the system that they chose. And that you can get to that solution, or that first solution, so to speak, or that, increased fluorescence through 1 mutation. And so it might be that the 2 copy populations are sort of you can rescue Ohno's hypothesis if the system depends, the two copy system or the system itself just depends on more complex mutational pathway. You've got to get sort of this set of mutations together in the right order at the right time for that new function to kind of emerge. There again, recombination being in the mix feels like a really important piece of the puzzle for Ohno to kind of come back into the game under the regime of duplicated genes outstripping the single copies.
I mean, I think it's a great simplified system, but having no recombination, I think, makes it very artificial in some ways.
I think that's right. And they kind of, they set that as sort of their marker of their study. Like that was kind of really how it was defined. And I think in that constraint, they sort of narrowed the window of what they were sort of able to get at. They do point out, I think it's, it's still like doing the experiment and kind of that back and forth between here's what we saw and here's what you think. It really helps to, I think facilitate that conversation. So then they bring up the notion of functional promiscuity. So, how often, so is the gene before the duplication happens, is it poised to take on new functions? And so it's kind of an awkward phrase, but being pre-adapted is an interesting, I think, idea in protein evolution. And as biochemists, we're trained kind of in the old lock and key model. Here's a lock. The enzyme is the key that goes in and unlocks that thing. It has to match up just perfectly. In real life, I think the notion is coming out or increasingly seen in a lot of experimental systems where the enzyme is flexible. If you make a few changes here, it might do a whole new job, or there might be metabolic bypass, all kinds of possibilities. And so I think, this sort of speaks to that for a lot of duplicating systems to gain new functions, sort of the work needs to be partially done ahead of time and not already super constrained, so that window of the dilemma where you just lose the function, if it's pre-adapted, the chance that it finds a new function through random mutation and selection increases. So there we are. That was our attack, or the author's attack on Ohno's hypothesis, with a little dilemma thrown into the mix and some interesting directed evolution on the side. They just looked at the green channel here. Future studies, they might select on green and blue in different ways. That could be interesting because it might get at some of these more complexities. Can you do both functions at the same time? If you make this change, are you less likely to be able to do blue? You can only do green, or vice versa. I think it really has the system has some potential to unlock some more complicated biology as they continue to develop it.
How far could you get from fluorescence to do something different? Like a protein that does fluorescence, what else could it do besides fluorescence?
No, I love it. Now we're talking. Yeah. I mean, and it gets you daydreaming now that they've kind of set up the parameters of the experiment, can you put on a different selective regime?
Yeah.
And, it's obvious to do blue and green since that's what it already does. But to your point, like, do you do a different color, yellow, red, whatever, or do you do a whole different function? Can you throw in a selection where you've got like a biochemical byproduct that is poisonous, and could the GFP detoxify that, right? If so, that's a really strong selection, because most of the vast majority of the population will be poisoned. But if you hit that rare mutation, those ones might come through, and they might, now we're really talking in terms of neofunctionalization.
Because I'm thinking of early in evolution, RNA binding proteins, right?
Yeah.
They eventually become polymerases because that first step, binding RNA, is the key, and then you can do other things with just a few changes. So
exactly.
I'm sure they're going to do that, try and look for some other functions that they could screen for and select for. That would be pretty neat.
Exactly. And picking instead of a GFP, picking an enzyme or a gene encoding a protein that already that might have that possibility. And so you can kind of, it's almost like you can sort of pick the rare. So the more difficult it is, the more rare it is, the bigger population sizes, the more time you need. Or if you pick something that's a little bit easier, just get more green. Right, then it starts to become a little more like, it feels like, okay, well, you got it to turn more green because you got a little more protein, easy solution, sort of short circuiting evolution almost, and everything in between. So I think finding that sweet spot, right, where it's not so rare that it's impossible, to see it through the lens of your experiment, but not so easy to get there that it's just, flipping one mutation on and you're there. Finding that balance would really, I think, potentially move this system forward in cool ways.
So Tom asks, what besides fluorescence, oxidation reduction function?
Yeah, I have to confess my knowledge on the sort of jellyfish encoded or whatever marine critter encoded fluorescent protein, this one, in terms of the chemistry here. I'm not up to date on that, but
he says, but because light is
yeah,
absorbed and electrons bounce around in energy states. So
yep.
some functions
Oh yeah,
associated with that, right?
absolutely. Yes.
I mean, you could imagine this might be a photoreceptor, right?
Oh yeah. So the actual, the, photoreceptor, of course. Yeah. And so how these critters, getting back to kind of the natural state of these fluorescent proteins, how these critters find themselves in low light conditions, and then sort of communicate with each other through or warn each other, or et cetera, by using the emission of whether it's throughout the sort of visible spectrum or beyond, into UV, et cetera, depending on how they're picking up on it. All that biology is playing out sort of in tide pools and in deep ocean settings as well. So, and that's a fascinating area of biology that separate, I would say, from this study. But yeah, that's really interesting.
Remember a couple of years ago at the giant virus meeting, we talked with a lady who had identified a photoreceptor in a giant virus, right? Photoreceptor gene, right?
Yeah.
So could a fluorescent protein change sufficiently to become that? That's the question.
Yeah, that's another one of these unicorn horns.
You could design assays to look for that. I think that would be fun, because that's the whole idea here, right?
Yeah.
In this paper, they look at the parameters, but the real question is, what kind of really different functions can you get?
Yeah, and that's out there, by the way. People do directed evolution on fluorescent proteins, different ones all the time, to try to get new functions. It's a really cool area of literature.
So we have a couple other questions. Want me to go through them?
Yeah, of course.
So way back when you mentioned bar bodies, Vanity said, is X chromosome inactivation completely random on a per cell basis?
Oof.
I don't know.
I don't think it's completely random, but approaching random, possibly. Yeah, that's complicated by an interesting biology as well. Great question.
Yep. Tom said, now I'm thinking about pseudogenes as a consequence of duplication and subsequent mutation. Sure. Right.
Absolutely right. And so, this is happening in our own genome. So if we look at, I think there's something to like maybe 7,000 processed pseudogenes that have been annotated in our own genomes. And then, so the question then becomes, how did those originate? So a lot of them were copied and pasted by retrotransposons, so-called line 1 elements. And so those are, every time an event like that happened, these were sort of tests of Ohno's hypothesis, in a sense, right? So we had 2 copies. If it was an exact copy, it could show up dead on arrival, depending on how it copied. But then that window was open for some amount of time. And then if we look at a lot of those processed pseudogenes today, a lot of them are so riddled with deleterious mutations, the notion of them coming back online, certainly as a whole gene, is almost like vanishingly low. However, could you hit a mutation that puts a start codon, a methionine amino acid, in the middle of that thing, and then it runs, and you could have a new gene function sort of grow out of that. Actually, that reminds me. So Ohno also, I think, is coined the term junk DNA for all of the
wow.
yeah. So one that still gets kicked around today and is controversial and sort of argued about by various. So anyway, so you've got all these processed pseudogenes, but if it's in that first moment, if it encodes all of that capacity, that's like, it's sort of sensitive to a mutation that could be beneficial or could take on a new function. And in fact, in my lab, we think we see that as it relates to species or lineage specific immune functions. We've been looking mostly in the South American monkey genomes for reasons I won't go into, but we see a case where there's a processed pseudogene It's been duplicated specifically in that lineage of South American monkeys, so we can date that back to like 30 million years ago. And then if we look at modern species today, some of them still have that intact entire open reading frame, which is a little bit weird. It hints that there's something like keeping it around might have some benefit. There's others that have lost it altogether. It's littered with point mutations. And so it's kind of off the record.
Mm.
And then there's other species that have picked up one truncating point mutation. They've picked up some other mutations, but that activates that as a pretty potent antiviral against HIV and other viruses that depend on the certain cell pathway, envelope viruses, to bud free and infect new cells. And so, yeah, I think the pseudogenes, this is like in our own genomes, for example, are represent this sort of process of duplication with all of these possibilities of neofunctionalization, and the constraints on it that this paper gets at. I love this. Yeah, it's really, really fun to think about this as we now can see we can see entire genomes, we can compare them, and we can do experiments. We can really pick apart some of this process in a way that couldn't happen back in the 1950s, for example.
Hey, MJ asks, is that the process used by FDA to trace an E. coli 157H to its origin? I don't know what you're referring to, but that would be whole genome sequencing, right? They just sequence the isolates and look for related sequences wherever they may have come from, right?
Yeah, that's right. And so here they're using, I forget the exact strain of E. coli. They're not using, one that we would be worried about for sort of, like food poisoning or related pathogenic reasons. This is sort of the lab rat of E. coli. But all the same, MJ, all the same technology, genomic technology to type these things is absolutely at play here. Just for all the way from asking sort of these questions from, semi-obscure Japanese molecular evolutionists, all the way up to, that salmonella that might be in our unpasteurized milk, from sort of the basic science fundamental question all the way to sort of the applied, how do we present it or prevent a disease outbreak, et cetera. It's all woven together here. Yeah.
Here's one for you, Nels. Tracy writes, did both amygdalae develop simultaneously, or did they evolve in a more complex manner over time? It
I love
seems like
this.
people are easier to hate than love.
There's a lot to unpack here, Tracy. But this is great. So, the reason I like this question is how the concepts that we talk about, at the level of biological organization, how much do the same concepts apply at different levels of organization, right? So we've been talking about the level of organization of the gene, and you just take a DNA sequence, you duplicate that, and then you ask about all the dynamics as this holds together. Here, we're talking about, left and right amygdala, and let me just immediately concede my neuroanatomy is terrible, but like if we think about left hemisphere, right hemisphere, right? Or just bilateral symmetry, the fact that we have a left hand and a right hand, we have two eyes. There's redundancy built into our body plan here, or into these structures of the brain. And by, thinking about that as a sort of adaptive way forward. So, obviously, if we're, you lose your left hand, but you've got your right one, that's pretty great to have that sort of functional redundancy there. And so, how that's encoded in our development is a whole nother question to try to connect it all the way to the genes. But I really love thinking about that. And not only like from the genetic level or the gene level that we've been talking about here, or the tissue level there, or the sort of body plan level, but also like the social, like the organizational level. Do we have redundancies built into our social structures? Do we have redundancies built into our, sort of political structures that can weather these, sort of catastrophic events, or add that redundancy? Certainly from an engineering standpoint, really important. And in a lot of cases, again, like thinking about maybe drawing back to the concept of recombination, 2 is not enough. Having 3, 4, 8, 12, like when you're flying on that Boeing 787, and sweating it, given some of the ways that those engineers have been letting down the airline industry, you want like a dozen backups before in that level of redundancy to sort of feel safe. And so I think all of this sort of hints at some of the same themes playing out differently at levels of biological organization.
From Microbe TV's perspective, never have a crucial file on just one hard drive.
There
Always
you go.
two minimum.
There you go.
Because if you put it on one, it's going to fail. I've had it
Yes.
happen many, many times, right? I buy these little portable drives.
Oh yeah. Well, this happened,
Right?
Vincent. This happened.
Yeah, there
At,
you go. So these I got,
there
these
you go.
little SC. Notice I have two of them
Yes.
because
There you
you
go.
copy a file to one, then you copy it to the other because the one is going to die. I guarantee
Ohno's hypothesis right there in
it.
your left
It's right.
hand. And that saved us at TWiEVO 100. We had redundancy in our, I think our HDMI cables actually allowed our broadcast to go off on time because we had a faulty cable, but the second one came in and saved the day.
Well, when we visited the Needle up in Boston, the
Mm-hmm.
BSL4 lab, right?
Yep.
They took us on a tour. Everything is redundant. They have a mechanical space above the lab where they keep copies of all the generators, all the tanks, all the machines,
Yeah.
everything that they need. In case something breaks, they have it upstairs. They don't have to wait for it to come. That's just
Yep.
amazing. Of course,
Agreed.
that ends up costing a lot of money, so you have to decide how much redundancy. And in a biological sense, it is also a cost, right, to carry extra genes. And the
Absolutely.
organism has to decide but obviously because gene duplication is so widespread, it's clearly beneficial. Right?
Absolutely. And not only gene duplication, Ohno also spoke to genome or whole genome duplications. So we see this in the somewhat recently in the ancestors of fish, of bony fish, teleos. But this is, and we see this in yeast. We see this in polyploid organisms. I think eons ago, we talked about strawberries that have something like 8 copies of all their genes. We've talked, so you see this in nature, redundancy playing out not just at the gene level, but at the genome, chromosomal level, aneuploidies in different yeasts, et cetera, and at the whole animal or whole genome level as well.
Yeah.
Yeah.
Cool. All right, let's see. We have 2 more questions. Tryon asks, did mutations reverse?
It was great. So yeah, reversions happen all the time. And so that's kind of what was in it was cooked into their engineering. Why did they use 3 mutations to have an inactivated second copy? It's so that as mutations reverse, you have to do that 3 times. And so that becomes nearly like, again, a different sort of flavor of redundancy. If you revert one, but if you've broken something 3 ways and you fix it 1 or 2, it's still broken. And so that in that case, it covers. They didn't, I don't think, certainly in the kind of main text, they didn't really speak to that, but that's a way of sort of navigating or negotiating Ohno's dilemma as well. That window might close if a debilitating mutation comes, but it might reopen if that mutation reverts and now it's functional again. And in the meantime, if you've picked up a couple other mutations that might somehow be beneficial, you can almost do these genetic leaps forward. So yes, and great question. That's exactly right. They reverse.
All right, Les says elephants have less cancer, so live longer. Does having multiple copies of cancer-fighting genes like P53 help it, or are there other factors?
Yeah, I think yes, and all of the above. So this is an interesting case. Another case, echoing on Tom's question on the pseudogenes. So elephants are known to have extra copies of P53, as less as you're pointing out, and these are pseudogenes. So they've been duplicated by the same mechanism that we were just describing before. The retrotransposons are copying and pasting them just by a genetic accident, by the way. The retrotransposons just want to copy and paste themselves. This is anthropomorphizing, but that's sort of the genetic mechanism. They make a mistake. They, instead of grabbing their own gene, their own mRNA, on rare occasions, they grab P53 or another gene, host gene, copy and paste that, and that's how you get that extra copy. And so there's Vinnie Lynch's lab, has been looking at this, describing this for a long time, linking it potentially to the Peto's paradox, species that live longer and are bigger, have more cells, more divisions, right? How do you grapple with cancer in those cases? And so there's also a lab here, Josh Schiffman's lab, who's taken one of these, one or several of these extra P53 copies and are trying to advance that. What if we were able to deliver this to some of our cancer cells? Would this reverse a cancerous state? There's a company that he's launched that aims to do this, and I think they're still in early stages. But but yeah, so I think that's, that's one possible mechanism. It probably doesn't explain or solve Peto's paradox for elephants. There's probably other stuff going on there as well that that people are interested in, but are trying to get their head around using comparative approaches, other approaches. But yes, this is absolutely an example, I think, of how multiple copies could, work in ways that could have, beneficial outcomes. So the phenotype here is resistance to cancer, whereas the phenotype for the study was like more GFP. But this is all kind of on that continuum of thinking about linking the genotypes or the evolutionary process to the phenotypes, the outcomes in real life.
That was a subject of a TWiEVO, right? The elephants and P53.
Yeah, I think we did a Vinnie Lynch paper back in the day.
Yes.
Exactly. Yeah.
Vinnie Lynch. Okay.
Yeah.
Renzo says the complex nature of life, of how genes play a role in life, still baffles me. Philip Ball wrote an interesting book entitled How Life Works. Just adds a level of complexity to the subject. Great read. All right.
Cool.
Thanks for that, Renzo.
Yeah, agree. Yeah.
All right, Nels. Want to do some picks?
Let's do this. I'll get the ball rolling since my pick kind of relates to what we've just been talking about. So I can't get enough of experimental evolution and directed evolution. Well, I don't think that'll shock you. But so there's a new book, or not a new book, second edition of a book called Experimental Evolution in the Nature of Biodiversity. It's sort of in pre-release. This is Rhys Kaysen, who's a purveyor of experimental evolution, pulling together kind of in not quite textbook form, like pseudo textbook form, but thinking about experimental evolution as an approach, how that relates to setting up these laboratory conditions and sort of courses of selection. How do we relate that to the nature of biodiversity, or of the natural settings, more complex biology all around us? So I think the first edition of the book, I guess it's a textbook, came out in like 10 years ago, and the second one's about to drop. I have to say the field in the last 10 years has gone bonkers in terms of all kinds of new studies, all kinds of different systems, not just microbes, but flies and fish and all kinds of stuff. And so really, if that's a topic of interest, this is a great resource, I would say. So that's
cool.
My science pick. How about you, Vincent? What's your science pick of the week?
So I have an editorial in Nature. Let's turn it on here. It's called Is Science's Dominant Funding Model Broken? Should one PI have the power to determine postdocs' careers? And actually, there's a poll associated with this, but it's got some interesting ideas here. And I want to point out that the picture here is
oh yeah, that's great.
from the movie Don't Look Up.
That's a good one. Yeah.
DiCaprio is an astronomer, and Jennifer Lawrence is his graduate student.
Yeah, that's right. Yep.
But basically, as we've talked about here, right? You get a PhD, and then you can do postdocs, and sometimes you do a bunch of postdocs. But for some people, you end up getting an academic job as a PI, and then you can set up your own research group, and then you will get people who work for you, and you will go through the same thing, and you have lots of people training for very few PI positions, essentially. And it's stressful, it is precarious, it's not always conducive to a normal life, I think, right?
Sure.
So the question is that the way to do it? And what other solutions are there? So there have been polls where they ask people, what else can we do? And reference 2 goes into that. They they looked at this poll and they said they looked at the structure and outputs of academic research. To their horror, the researchers uncovered a system that ends up rewarding administrators and empire builders, not creative scientists engaged in research and mentoring. And I've kind of noticed that myself over the years. First of all, we had a lot of administrators at Columbia, way more than PIs, and I always thought that was weird, right? I think you'd want a lot of PIs to increase the chance that you're going to get something good. And then there are the empire builders, which is basically people who have many, many, many grants and 50 to 100 people in their labs, which I never thought was very productive, right? So that's one thing that's interesting. The article goes on to talk about this issue and how to deal with it. There's another model which is used by DARPA. In this system, grant managers picked from a pool of some of the most successful PIs wield considerable power and influence, but this really doesn't have any improvement over the PI
Yeah.
models.
Yep.
There are other models too, but and they want you to put your thoughts in here in this poll. But that's
Cool. Yeah.
I spent my career, doing the traditional thing. I got a PhD, I did a
Yep.
postdoc, and then
Yep.
I got my own position, and then I trained people.
Yep.
Some of whom, let's see, my postdocs, most of whom went on and got some kind of position,
Yep.
right? Which is rare because many postdocs do not. And so, just because it's something we've always done doesn't mean it's the right thing. But if you're going to
Yeah.
change, you need to have a good outcome. I don't know if you've ever thought about this, Nels,
Oh,
but
of
you're
course.
doing
No,
the same thing. You went through the same
yes,
process, and now you're
that's right.
training postdocs as well, right?
Absolutely. No, I mean, it's funny. It kind of echoes, maybe getting back at the layers of organization, echoes on this paper, right? Where here, the selection is this system where, you go through all these hoops and to get grants and to get
Yeah, yeah.
fellowships and to go and apply for jobs. That's the selection. That's the equivalent of the fax sorter. And so, the more rigid that selection is, the more you either have cheaters who are gaming the system or doing it in ways. You have more administrators, you have the empire builders, which, that's one method. But if we're selecting just on empire builders, think like that kind of squelches out some of the grassroots energy or creativity maybe that they're pointing at the beginning of that piece. And so I've done a little bit with DARPA actually, or not, I haven't been funded through any DARPA mechanisms, but I've like applied for one or two and seen how messy that can be. That's a whole nother sort of selective scheme that I think can go off the rails, as they point out too, pretty quickly. But I think maybe, I agree. The more we're talking, first of all, talking about this, secondly, building more paths that are relaxing the constraints or the thresholds so that there are pathways forward. Also, again, the equivalent here, or the analogy of phenotype. Let's be careful not to, describe whatever
Mm,
adaptation being more GFP or being more PIs and academics, right? I mean, that's one
yeah,
of the, I
yeah.
think, the really good things happening right now is that we're seeing people training in science like you and I practice, Vincent, that are now going on to great careers across the board, where someone could be a great successful PI in academic science, or it could be a great successful scientific career in biotechnology or in public policy or in patent law. And I don't see one as inherently better than the other, just different
Yeah,
kind of
yeah.
pieces of the puzzle. Yeah. But oh man, yeah, the pressure, the selection is strong, as the title points out, the model being broken. Yeah.
Well, the academic PI is not the only way to do good science either, right?
Yes.
Like, it's a
Exactly.
very
Yep.
appealing thing because you have a lot of freedom to look
Hmm.
into what you want.
Yep. Yep.
You can train people, which is really nice. But there's government laboratories. There are
Yeah.
pharmaceutical private company laboratories, and in addition, in many places like the NIH, there are staff scientist positions, which are not PIs per se, but you can have a small research group under someone else. And for many people, they're happy to do that. So
Yeah.
not, not everyone needs to be a PI. I guess that's the point.
No. Yeah, and actually that's where I think we could really do better quickly and kind of easily in academic science, is this just to solve the staff scientist position. So it's just rare, right? That I mean, I think both you and I have had some of the core discoveries. There's the lab manager of the LD lab has not gone through all of the rungs. She got her Bachelor of Arts degree, Bachelor of Science degree, didn't get a master's or a PhD or do a postdoc, but is making the coolest discoveries in my lab. I mean, is really the whole fabric of the lab, holding it together, organizing it, etc.
Right.
But
Right.
it's kind of rare that we're able to that people like take on that role or are able to be supported, and it's because we aren't designed that way. We don't have the resources set up. If the NIH style of staff scientists could play out to the universities across the country, if universities could get their act together and dedicate some resources, not just to those administrators, the sort of associate dean of research whatever weird sort of thing that keeps mutating and growing. But instead we had 4 staff scientists funded by that. Like, now we're talking like that. Yeah, that would be fun ways to experiment, to break the dominant
the point
funding
here,
model.
folks, is that staff scientists would have to be supported by the university. That's the whole point. Because if a staff scientists were, let's say, supported by Nels, there's no security, right?
Yeah,
because
that's right.
if he loses his grants, then the staff scientist has to go. But if you work and are paid for by the university, that's not the issue. And that's the whole point about a staff scientist, that you don't have to worry about raising money.
Yep. And honestly, like if a university made a strategic decision to cut 3 administrative positions and add, let's say, 30 staff scientists, I think the science that would come out, meaning the indirect costs that would come from the output, it would take a few years, but would more than pay for whatever perceived advantage there is in that sort of growing administrative structure. So that's I think there's all kinds of low-hanging fruit here, competitive advantages. If universities were forward enough looking and wanting to do some experiments along these lines, could it outcompete their peers by moving some of these resources around like this, instead of building another building, setting up funds or endowment, or using some of the endowment, et cetera, to fund more staff scientists. What a great way to attract talent and keep it.
But yeah, we have way too many administrators. It's just crazy. They have just multiplied over the years that I've been at Columbia. Oh my gosh, I just don't get it. And Noughts and Crosses says universities getting their act together. Oh yeah.
Oh yeah, no, I'm not. I'm not Pollyanna over here.
All right, that will do it for TWiEVO 103. I want to thank everyone for being here, particularly our moderators. Barb Mack, UK was here. Vanity Nutrition was here. Les was here. And Tom Steinberg was here. Thank you all for moderating. Don't forget, we have office hours tonight, 8 p.m. Eastern. That's another one where we haven't done for a while. Hoping to get that going again. That'll be right here at YouTube. And you can find the show notes associated with this episode at microbe.tv/TWiEVO. If you enjoy the work that we do here on TWiEVO and all the other Microbe TV podcasts, we'd love to have your financial support. You can go to microbe.tv/contribute, or you can give some money at Venmo. I can't point. Nels, you have to point over your head.
Sorry. Yeah, let me get it
Venmo.
right there.
Yeah, Venmo. There you go.
So,
you can.
Yeah, this isn't subtle, Vincent. We've been, if we could get like the emoticon sunglasses with Venmo across there, that's going to happen. That's our next. Yes, to underline this, your support is so important to us, and you being here is so important. So in addition to our moderators, thanks to everyone else who came. We kind of scrambled the calendar, but everyone showed up, and more people showed up. And the questions, as usual, the comments have been so fun. So thank you all for your contributions.
And, we're back on a regular schedule now. So once a month, you can. Probably at about a month from now, we should be doing another one. But it's always on the Microbe TV calendar. What is it? Microbe.tv/calendar. Nels Eldey's at cellvolution.org. Early Bird on X. Thanks, Nels.
Yeah, thank you, Vincent. Great to be back together. Great to fire up LD Lab Studios, and can't wait for the next one.
I'm Vincent Racaniello. You can find me at microbe.tv. Music you hear on TWiEVO, let's get that ending music going here, is by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.